Hello and welcome back to the channel. Today we want to talk about a biochemical technique, which are Western blots. Those are actually an analytical technique to detect proteins within samples. Now, it was invented in 1979. First you have to separate the proteins according to their size, and then you can isolate them. You have to transfer the proteins to a solid basis, and you need so-called secondary antibodies to visualize them. So Western blot get used for proteins, but there are also southern and northern plots. Southern plots are used for DNA, northern plots for RNA. The original paper which described this technique was cited as much as 400,000 times, and I guess this highlights the importance. It's combined routinely with a technique called PAGE, which is polyacrylamide gel electrophoresis. And we'll talk about that in a minute. It's a technique to separate proteins. Now, actually, you have 6 steps required for performing a Western blot: sample preparation, gel electrophoresis, protein transfer, blocking, antibody incubation, and protein detection, as well as visualization. The proteins can get extracted from different samples, which can be tissues but also cells. As soon as the proteins are extracted, it's essential to determine the concentration of the proteins. And this is required to know the amount of probe to be loaded. And you can estimate the concentration, for example, via a photometer. Now we want to talk about PAGE. It can separate proteins ranging between 5 and 2,000 kilodaltons, according to the pore size. And the pore size gets controlled by the concentration of the gel. So you have to choose the appropriate percentage of the separating gel. The rule of thumb says that the smaller the known weight of proteins is, the higher the percentage of the gel should be. As soon as the proteins are separated, they are moved from within the gel onto a solid support membrane to make the proteins accessible for antibody detection. The main method, actually, for transferring proteins is called electro blotting, and this uses an electric field to pull proteins out of the gel and move them into the membrane. And for this purpose, the membrane gets placed between the gel surface and the filter. And here you see the general principle of PAGE. The bigger proteins cannot move through the gel as quickly as smaller ones, as the gel is like an obstacle for the proteins. So the smaller you are, the easier it is to pass these obstacles. So then you have to place the membrane in the blocking agent. And this is due to the fact that the proteins attach to all the places in the membrane where the target proteins have not bound. So blocking is used for avoiding unspecific binding. Then the primary antibody binds to the target protein. As soon as the primary antibody, of course, gets incubated with the membrane. And the choice of your primary antibody depends on the antigen you want to detect. It's usually derived from animals like rabbits or also goats. And then you can wash the membrane with an antibody buffer solution to minimize background and also to remove unbound antibodies. And there are also a lot of washing steps. And then you can expose the membrane to the specific enzyme conjugated secondary antibody. So the secondary antibody is labeled and can bind to the primary antibody. And the primary antibody has already reacted with the target proteins. So based on the species you used for creating the primary antibodies, you choose an appropriate secondary antibody as well. Then the substrate can react with an enzyme that is bound to the secondary antibody, and this then generates a detectable signal. This is of course highly important. Horseradish peroxidase is, for example, commonly used. So this is a technique that is quite often performed within labs, and it's actually a standard procedure. I hope this was a quick overview, as it's important to understand the principle behind it. And if you like this video, don't forget to give it a thumbs up, but also to subscribe to the channel to see weekly uploads on highly relevant topics in medicine, in mathematics, biology, and of course biochemistry. See you in the next video.